Hi, everybody. Welcome to another snow day edition of Bio 106 Lab. So this week we're going to be talking about enzyme kinematics. Basically, every function in a living organism is mediated in some way by enzymes. So these are a big deal in the field of biology, and the way they work and what affects them is something that is crucial to understanding how living organisms work. All right, so I hope everybody's doing well today. Hope you're feeling well and maybe resting up on this snow day. All right, so let's get. All right. So enzymes take part in two different kinds of reactions. Exergonic reactions, which are the breakdown of larger molecules. All right, so for example, metabolism. If you've got enzymes that are involved in breaking down, say, sugar molecules, that is releasing energy. Right? That's meaning that you can then utilize that energy. And then the other one are endergonic reactions, means that you're building large molecules from smaller ones, okay, putting things together. If you want to go with the Lego model, exergonic reactions are taking Legos apart. Endergonic reactions are building things with those Legos. Right? So the endergonic reactions use energy, and they're making larger molecules out of simpler molecules. So most reactions require energy input. This is called the activation energy. It's just the analogy is that there's a kid on a slide that just needs a little bit of a push, and that will get the reaction rolling. Okay. What enzymes do is they lower that activation energy. So without those enzymes, you would require too much energy in the form of heat. Right. So most of the reactions that are happening in living organisms that require so much heat that those organisms couldn't survive the amount of heat that's being produced. And so basically most metabolic reactions would never happen unless you've got some sort of enzymes to mediate that reaction. So basically you can see energy of activation with the enzyme. This, if we're sticking with our child on the slide analogy, this energy of activation is that little gentle push that you would need to send the child safely down the slide. Okay, whereas normally what the reaction would take is a much higher energy of reaction, and with that same analogy, that would mean standing at the bottom of the ladder to the slide and just heaving the child over it. Okay, one is much easier and much safer for the organism involved. So enzymes act in two different steps. Right? So first, the substrate is going to bind to the active site of the enzyme. And keep in mind, these enzymes are specific to substances. So each enzyme has its own specific substrate that it will react to. And then products are formed. Okay, so in this case, we are taking a substrate that is a single molecule and breaking it into two molecules. So enzyme binds to substrate. You briefly get an enzyme substrate complex, and then the products are then released from the enzyme. Right? So some big things to remember about enzymes. They're all proteins. Okay, they're acting as biological catalysts. They are facilitating everything in your metabolic pathway. They accelerate the conversion of substrate to product, so making reactions happen fast enough that organisms can continue to live. They're very specific. They'll only react with one or a limited number of substrates. All right, so enzymes don't alter the reactants or products of a chemical reaction. So basically, they're just facilitating the reaction, getting things going. They're not doing the actual alteration. And enzymes are not altered by the chemical reaction that they're involved in. So that means you can add enzyme to a reaction and it will continue to react as long as conditions are as long as conditions are good and as long as there's still some substrate to react with. And our enzyme names are usually named for the substrate they act on with the suffix ase, right? So, for example, silobiase. So enzymes function in a narrow range of environmental factors. So the big ones we're talking about is temperature, pH, and salinity. And so, for example, with temperature, in most cases, and this is no exception, reactions will happen faster as more heat is added, and that will happen up to a point. So this, you can see that this enzyme has an optimal temperature, right, where it will react the fastest. But extreme high temperature will make the enzyme non-functional. Right, so you can see after that optimal temperature, the functionality of the enzyme starts to drop and then precipitously drops off. That's because what happens to the protein is it denatures. Enzymes, all right? Most enzymes in living organisms function at a pH just above 7. All right, so for humans that makes sense. We're right around 7. Our blood pH is right around there. So what'll happen is, yes, it'll function outside of that range. So you can see, this one here, even down to pH 2, which is very acidic, you still see a little bit of function there. But there is an optimum range where this is going to operate, right? Temperature, that varies on location, so where the enzyme is, and that includes what organism it is in. So it's not surprising that organisms like humans that operate at about 37 degrees Celsius, that their enzymes optimal range is about there. Right? So if you've got animals that are operating at lower animals or other organisms that are operating at lower temperatures, you would expect that their enzymes would also function at those lower temperatures. So how do temperature, pH, and salt affect the actual molecule, and so affect its function? So enzymes rely on chemical bonds to remain intact. So high temperatures will disrupt the hydrogen bonds. That causes a lot of unfolding in the protein. And remember with proteins that their function is based on their structure. So if you change how they're folded, you change how they work or whether or not they'll even react. And then high or low salts or changes in pH outside of a certain range, those can disrupt the ionic bonds that are in those proteins. And again, leads to denaturization, causes it to change its shape and no longer function. Right? So if you disrupt those chemical bonds, you end up with a denatured protein. So in the lab exercise, the enzyme we're looking at is I forget what the O is, but 6-O alpha L, it might just be O, raminosyl D-glucosidase. All right, so looking at that name, we would think that it has probably something to do with glucose. All right. So our substrate that we're using is starch. So this is a complex carbohydrate. All right, it's made these long chains, and this is what the enzyme is going to react with. Right, because starches aren't utilizable in their starch form, in their full long chain form. What you need to do is break those down into simple sugars, and that's what this enzyme is going to do. Then we have a stop solution. It does a couple things. One, stops the enzyme from reacting with the substrate. The way it does that is generally there'll be a very high pH. And so the enzyme will stop reacting. So that means that you can basically take a snapshot. Right? You can say, okay, at this time, we let it react for this amount of time. We add our stop solution. It stops everything. So what we see in that reaction is what was happening at that, what had been happening at that time, right? And the other thing it does is it changes the color of the substrate. All right, so anytime you've got simple sugars, these simple sugars will turn from a clear color to a yellow color. All right, and so the more yellow the color, the more product that was made. So if you had put these enzymes in and put the substrate in and then added the stop solution, and if there was no reaction, you wouldn't see any yellow color. There has to be reaction and there has to be product in there before that yellow will show up. So the more yellow you have, the more product that was made. Right? The more the enzyme broke down starch into simple sugars. And the more yellow, the more optimal the conditions were for the enzyme. And the way we measure that yellow is we are using a device called a spectrophotometer. And what that's measuring basically is the amount of yellow. Right? We put light through it at a wavelength of 450 nanometers, and we record how much of that light was absorbed. We're using 450 nanometers because that is the one that is most absorbed by the color of our yellow, right? Our product that we have. So what you guys are going to be doing, we're going to combine the enzyme and substrate under varying conditions, temperature, pH. You're going to add the stop solution to the reaction after a set time period. You're going to analyze the color of the solution using the spectrophotometer. Remember, more yellow means more reaction. And you're going to compare the amount of product made in different conditions. I'm going to give you a raw data set that you'll be using to write your paper from. I'll go over that in some more detail. Plan right now is I'm actually going to put together a whole nother video and talk about the data, talk about how to process it, how to graph it, all that kind of stuff. You guys need to think about what you need for graphs. Right? So how many graphs are you going to do? What are you going to graph? Okay, that sort of stuff. What kind of statistical tests are you going to use? All right, are you looking at two different sets of data and comparing their means? Then probably going to want to do a t-test. If you're looking at more than two sets of data, you're probably going to want to do an ANOVA, that sort of thing. So what's your null hypothesis? All right, for temperature, null hypothesis is that temperature has no effect on your speed of reaction in your enzymes. Okay, so then what would be the alternative hypothesis there? You think about that. What's the question you're trying to answer here? Right? What are we trying to find out about enzymes? So what you need to complete for next week, is the pre-lab. The pre-lab quiz, right? So that is the same as what we would have done if we were in lab. Then go through the makeup lab, and you're going to complete that and submit it on Blackboard. And then you can get started on Write Up 2. So remember, like we said above, I think I might have jumped over it. Is that for Write Up 2, you're going to be doing the results and the discussion. All right, so you're going to be focusing on making sure that you're doing the right graphs, describing your data properly, and you're going to be doing the discussion, which is sort of wrapping everything all up and talking about what your results actually were. So there's going to be another writing workshop on February 26th. I will also be available, office hours, email, anytime you need to talk about any of this stuff. If you would like me to, I can go over drafts. Okay, so if you email it to me, I will go over a single section. So either your results or your discussion. If you schedule time with me and come into office hours, I will go through all your stuff. Okay, that's the deal. That is pretty much it. All right, until next time. So hopefully I see you guys next in-person lab. Again, if you have any problem, any questions, just let me know, email me, or come to office hours. I hope everyone's doing well, and I will see you next time.